they are on hot, they are at the end of their program. And that means that they are making this all-out push to submit. There she is.
Hello. Yeah. And
so, but thank you for coming. Welcome to you.
I was trying to get Dr. up here, but she said she's on her way now.
Yeah, I was wondering where she was.
She said she's on her way now, so
okay.
I tell her to go out as long as we can. But I guess we ready now, right?
Yeah, that's good.
So. All right, folks. Good afternoon. I'm trying. Good afternoon. I'm inside. Okay. Well, I'm Arthur Potts, for those of you who don't know who I am. And I'm part of that program that Professor Stout just mentioned will be starting very soon, the Marine Sciences program, the Bachelor's and the MSc program down at the Chagaramos campus of the UTT. Right, this gentleman here is who have us here today because he has been doing some work in our country on the coast of Toco, and we kind of accidentally ran into each other somewhere online, and then he told me it's because somebody at Belford University, where I attended some years ago, told him that if he's going to be down there doing this research, see if he could get a chance to meet with me and so on. So he communicated with me and said that he was willing to present his work as far as he has gotten with it to us if we could put together a session for him. So I, of course, contacted the graduate, the professor responsible for graduate studies, and Professor Stout quickly agreed to do everything she can in short order and explain just now why we don't have the room full and Marshall, we don't have standing seats and so on. Alright, but the bottom line is that I thought this guy was American who had snuck into our country and was doing some work on some holiday period until the professor he has to sell his bio and then he realized he was from America. But it is good. He studied, he did his high school work in Arima and went off to the US on a scholarship and got a bachelor's degree at Martin Methodist College in Tennessee. Stanton Belford, as you see his name there, not Stanford, that is spelled on one spot in the advertisement. He then earned a master's degree in biology at Middle Tennessee State University in Murfreesboro. At MTSU, he successfully completed his thesis entitled Analysis of Coral Distribution and Coral Symbionts in a Patch Reef on Fringing Reef in the Southern Caribbean. All that is out at Toco, right? Enjoy that. And then he continued doing work abroad with the Auburn University, folks making arrangements for him to have research money spent on him so he could go and do work in the Red Sea and Key Largo, Florida. He then got some money recently from the American Museum of Natural History, Learner's Grade Marine Biology Award 2013, and that helped him to continue doing his work at Celebia Bay in Toco. He's currently a PhD candidate in the Department of Curriculum and Teaching, Secondary Science Education at Auburn University. And a PhD candidate at Auburn is a person who would have completed all the coursework, passed what is called a preliminary examination, up to a series of exams that all the committee members one day each are able to do. Once you pass each of their exams, then your major professor signs off with the committee members that you become a PhD candidate. At that point in time, you need to do everything in your power to have a successful dissertation, otherwise you'll have no degree. But he is at the point where all he has to do is complete his dissertation and we'll have a brand new Trinidad and Tobago citizen becoming a PhD that somewhere in the world we might be able to ask him to come back to help us at some point in time. So without any further ado, let me hand over to Mr. Stanton Belford, who's going to talk to us today about liming next to coral reefs, no drinks allowed somewhere there to go in every start.
Thank you, Dr. Potts. Thank you once again for taking time out of your schedule. What I've realized along the way in this entire process is that there's a lot of red tape you have to go through to share knowledge. When I began my master's at Middle Tennessee State University, I was in the middle of Tennessee, and a lot of my students asked me, how are you going to do research in coral reefs? But I'll take you on a journey, an initial journey on why I chose this, and try and connect with you some of the current data that I've collected, and myself co-authored with Dr. Philip from Life Science in Marine, and we published 2 papers actually on our findings here. And actually, last week when we went out on the reefs, the research that we're doing now for future research is actually looking further out on the fringe of the reef. So I'll take you to some of that data. I'll also take you to some snapshots of the biodiversity of the Toco reefs, because it's really a triangle under the sea. And the most beautiful thing about that is that it's less than knee-height water. You're finding this biodiversity during low tides. So the entire my goal really in life is, believe it or not, to share knowledge with students and teachers at Toco campus. That was my initial goal. I'm still working on that goal. Initially, when I took photos of the reefs here at Toco, and I would take it back with me to show my friends in Arima, they would say, wow, these are so pretty. Where's this in Tobago? And when I tell them, no, it's Salivia, or Salivia, to get the right terminology, they couldn't believe it, because they know they go there, they go on excursions there, they just go on a leisure trip, and they party, they eat, they're climbing right next to the reefs, but they don't realize that what's under there, the human environmental impact is really becoming more and more researched. With 7 billion people on the planet, let me tell you, ladies and gentlemen, we are making an impact on the environment. We have a local resource here that needs protection. We hear all about Toco Reef, but we hardly hear any protection of the only fringed reef in Trinidad, which is at Salivia. So that's why I came up with the line next to Toco's Reef, because I'm sure most of y'all have been, you all know the Kishon Walter Toco Lighthouse. Salivia is just on the left there. I'm sure most of y'all have been there, and y'all have lying there, relaxed there, but don't realize I'm hanging out next to the reefs. But the no drinks allowed part is, and I'll share this with you, some of the detrimental impacts of what we do there. So, why do research on Toco reefs? This actually began from a high school teacher in Arizona Senior Comprehensive, Ms. Carol Jericho, taking her class, her 6th form class, to the reefs at Toco. So these are, if you notice, the date 93. So she took us way back then. And you see my little scribbles here, there's sea urchin, hermit crabs, sponge, there's different mollusks, chitin, what we know as paco. So we did like an investigative survey of the reef. This is in 93 now. Fast forward to 2006, where myself with longer hair back then, doing now the thesis, a master's on reefs at the same place where high school teacher taught. So don't you dare tell me that a high school teacher can't make an impact on your career. I've been fortunate to complete a master's on the reefs at Toco, and this is next to the fishing depot at Toco, just to the right. So most of, a little bit of data that you'll see today is actually from here. I have 66 log scuba dives in the Red Sea. I have 23 log dives in Key Largo, Florida. So you cannot tell me that a high school teacher cannot impact your life, because it has. It has directly placed you on a career, on a marine biology career. So that's kind of like the story from the high school to the master's, and it's, still today, I've been fortunate to travel around the world are very much it, doing stuff like that. So, coral reefs, all right, you have three main types. You have the fringing reefs, the barrier reefs, the most famous one is the Great Barrier Reefs. You can actually see that structure from space. And you have the atoll, right? The fringing reef basically is close to the shoreline. The barrier reef is actually separated. The shoreline and the reef is actually separated by a lagoon, a deep lagoon, sometimes 800 to 1,000 kilometers offshore. And then your atoll is actually outcrops of reefs on a mound. In the Pacific, they actually are on submerged volcanoes. These reefs, the atolls, are actually, the water around it is much more pristine because there's no human impacts from rivers or close by, but as fringing reefs. There is a barrier reef in the Caribbean, and it's off the coast of Belize. Right? Most people know barrier reefs, Great Barrier Reef, and that's all they know, but there is actually one in the Caribbean. So for the purpose of this talk today, we're actually looking at the only fringing reef in Trinidad, which is at Olympia. So this photo I took last week, Wednesday. This is actually, if you could dim the lights a little bit, but this is actually the fringing reefs at Olympia is actually extends 200 meters from the shoreline. So
I would like to say, no more deal. We can't alternate lights in the street. That's not switch.
That's you're ready to see. So this is actually at 180 meters out on the reef during low tides, looking back at the shoreline, which is really neat because reefs in general, they reflect. All right, as you go towards where the waters break on the reef crest, you find you get a lot of much more diversity out there, and then the reef goes down into the reefs. There's a lot of species diversity out here. And I can tell you that every time, since I've been doing this 2005, beginning with the masters, every time I go out there, I see something totally different. And I'm in awe, my eyes light up. I bring my camera, I take 100 shots of one specific organism, just to see, hey, we saw this at this particular month, this particular year. Some years you see a lot of octopi, some years you see a lot of sea slugs, but it's a record of biodiversity. Because at Owen University, we just built our new biodiversity building. And so one of the things they're doing now, they're shifting towards photo, their museum is shifting towards photo records, digital records. So instead of stacking a lot of stuff, they're asking citizens to take photos and turn it into them latitude, longitude, temperature, location, so they have a record of what's there. Because that's how we can track different things that at least a long term, we can track biodiversity in that. That's one of the things I am doing there as well. So, coral reefs, there's a lot of, it's so complex, but there's a lot of interactions that are taking place. In any coral reef course, one of the first things, or first words you'll hear is symbiosis. Right? Symbiosis, you'll hear two main words when you're dealing with coral reefs. You'll hear nigerian, which basically means stinging, because corals, sea anemones, and jellyfish, they sting. Some sting more worse than others, and our skin is thick. So if you hold a sea anemone, it won't sting you as much. But if you hold it for 2 hours, and you wake up the next day, and you feel your hands numb, it's because it was actually stinging you. I know this because when you weigh sea anemones, you have to sort of do this back and forth so that as much as the water drains out, and then put it on the scale. And then I wake up the next day and my hands are numb, and I'm wondering why they're so numb. It's because it was actually stinging. So symbiosis, what is symbiosis? Symbiosis is basically a close association of two or more different species. Right? So there are actually 3 types of symbiosis. you're mostly aware of parasitism, to some extent commensalism, but with respect to corals, they're mainly mutualism is what we're going to basically look at. So how do we define these? Basically, you have species 1, species 2, and parasitism, there's a one-way interaction with one species 1 benefiting, but at the same time species 2 not benefiting or even harmed in the process. And so that is parasitism. In the case of coral reefs, in the last 30 years, we're seeing more and more coral diseases. If you want to go into some really hardcore genetic work, this is actually one of the areas to go, because there's a lot of research that's been done, but at the same time, we still don't know exactly what is causing some of these diseases. So when I say coral diseases, black band disease, yellow band disease, different things like that. In this case, it's black band disease, because you can see that black band, or that black ring. And it's actually, I think with this, there's white band, black band, yellow band. There's actually a concoction of bacteria that's actually attacking the coral, and literally, you know, the bacteria, they're benefiting, and the coral is being harmed. So that's one instance of parasitism. For commensalism, right, there's, once again, species 1, species 2. There is a one-way interaction with species 1 benefiting, but at the same time, species 2 is not benefiting and not being harmed. So that's commensalism. One of the, I guess, more well-known example is, you know, these birds sometimes you see in the field, they eat grass on the cow's back. In this case, so the bird will pick off the little parasites from the elephant. And a lot of people will say, well, the elephant is benefiting from getting a cleaning, but at the same time, you know, it's like, well, maybe the elephant is not really benefiting. So it's just a one-way, but the most important thing is that species 2 is not being harmed in this situation. One of the cool examples I learned this year is this particular African vine that actually uses a tree to climb it and get to the canopy and put out its shoots to get its nutrition as well. The tour guide told us it's not really, it appears like it's squeezing and harming the tree, but it's really not, it's just using it for leverage. So that's another example of commensalism. In terms of mutualism, we have a two-way interaction. Species 1 and species 2 benefiting from the interaction. And one of the most famous, when you think of mutualism, other than you think of corals and you think of sea anemones, but one of the well-known examples is the acacia tree. I know you all would probably think of a bird eating a fish, no, that's not mutualism because the bird is dying, right? That's more predatory. It's the acacia tree. The acacia tree has these long thorns, but at the base of the thorn is a tiny little sap, hollow nodule with sap in it, and the ants that goes on the tree, they actually make homes and eat the sap. And at the same time, if other animals come and eat the leaves, the ants actually bite them. So it's like benefiting the acacia tree, providing a home, plus a little nutrition, and the ants are like attacking and keeping away the extermination. So not the kingfisher and the fish. That's what I was trying to throw you off. But I was expecting more students, but that's fine. All right, we've all heard of Finding Nemo. A lot of people don't realize that there are actually 29 species of clownfishes. So this is actually one where we worked in the lab. It's the two-band clownfish. And this is the one I studied in the Red Sea because it's only found in the Red Sea, it's endemic to the Red Sea. So this is the clownfish, which depends on the giant scale anemone. These are actually juveniles. I've seen, this is Entacmea, scientific name, Entacmea quadricolor, and I've seen these in, so when you work in a lab, you see them small like this, and then when you go in the field, you see them like huge, oh my gosh, and your eyes are like bulging out because It's so cool. But obviously the anemones, they keep predator or larger fishes away by stinging them, and at the same time the clownfishes, they pass on nutrients to the anemones. And actually I've seen clownfishes don't grow past this size here, and I've seen these fish like really attack larger fishes as well. One real cool note about this is kind of like a cash cow question if you're in New York and you're jumping a cash cow. The larger of the pair of these clownfish is always female. So in Finding Nemo, I always break these kids' hearts in the middle school when I go and talk about this. In Finding Nemo, when Nemo's mom died, Nemo's dad, Marlin, should have changed into Nemo's mom. Because that's what it's called pertanders. They're pertanders. So what would have happened? Marlin would have changed into Nemo's mom, and then another clownfish from somewhere else would come in. So that's kind of crazy because you're thinking, oh my gosh, it's what, male change to female? Actually, this is common in the ocean. Fishes do it, Rosses do it. So there's a lot of sex change going on there. You should see them at the schoolers. And since I have your attention, you know Marlin swam the ocean, swam the EAC to find Nemo. Well, in real life, the anemone is located here. The clownfish, in all its life of survival, will only swim from here to here and back. So, don't believe everything you see. But you can't say that Finding Nemo because everyone loves Finding Nemo. Alright, so the trying to get some actually. You could actually play anything on clownfishes, even believe stuff goes on. And by the way, we still do not know what, so we know that anemones sting other fishes, but we scientists still don't know for sure if the clownfish is immune to the sting, or if they develop a coat, or if, we still are not sure. I just threw in this photo here. This is actually a Caribbean sea anemone. It's called a corkscrew sea anemone, and it actually has a symbiotic relationship or mutualistic relationship with this is a Pericles shrimp. And the cool thing about these shrimp, and we have these in the tanks at Walton, it's a mutualistic relationship. But if you want to get a pedicure, you stick your finger into the tank, and then if the shrimp signals you with the antennae, then that means it's ready, and you put your finger closer, and it will jump on it, and your finger can actually give you a pedicure. On coral reefs, you have these stations called cleaning stations. So, and you actually see fishes, like literally, like lining up to be cleaned by the shrimps. And you actually see this in Shark Tank, the movie. It's amazing how movies kind of show you you can teach some science with movies. So I thought it was good. Sorry, man. I'm sure if you stick your feet in there, you'll get it. All right, so we're going to focus on, we're still studying symbiosis and mutualism. So corals are animals. Believe it or not, in the late 1800s, scientists actually thought they were plants. But they're not plants, they're actually animals in the phylum area. Within the coral animal, within the cells, actually the middle layer or the mesoderm area, they house tiny dinoflagellae or algae or protists that's called zooxanthellae. The zooxanthellae, they are very important because they are photosynthetic organisms. So they use sunlight to make their own food. This is why you do not find corals or coral reefs past, or any Nigerians past 200 meters because they need light. So that's very important. Now, there's so many, there are at least Scientists have estimated at least 6 million zooxanthellae, tiny cells within a square centimeter. So we have a lot in a space. So the mutualistic relationship is that these zooxanthellae, they're given a safe home, and the zooxanthellae, they actually release very complex amino acids, phosphates, nitrates, and those sort of seep in to the coral animal to be used as well. Right? So these are very important interactions that take place on the reefs. So to get into biodiversity of the reefs at Toco, Trinidad, which I started way back when, but it's always amazing that if you, as a field biologist, I rather be in the field. That's part of it. That's actually the easier part of the job, is when you come and you have to enter data and you have to sit in front of the computer, as professors will tell you, all that's the toughest part of the job. So what we did as part of the methodology, we decided I initially used quadrats, right? And then I used the line and point transect method. So basically it's, I took a tape measure and 2 people on each end, and initially I started every meter. So this is actually some of the work we're doing now at Salavia, but instead of doing, so I did vertical, some of the work, the results of the work you'll see here is actually vertical transects. But because we wanted to highlight more of the upper in the tidal reef flat at Salavia, we went horizontal, because you get more distribution there. And so what I'm doing here is basically looking at every 50 centimeters out of a 100-meter tape measure, and just basically taking distribution, abundance, and diversity as well. All right, so some of the things you actually see, you see a lot of algae. This is green algae, phylum chlorophylla. So this is actually, what do they look like grapes, right? So this is actually green grape algae. Scientific name, Colourpa racemosa. So as you get towards, I know the program is initially starting coastal management, as we get to this here, almost everyone, professors and students, scientific name becomes very important, because these could be called something different in Antigua, or in Barbados, or in Jamaica, but the scientific name will always be that. So that's a crash course right there. That's necessary in the marine biology aspect. So these, because they look like feathers, they're green feather algae. And these are really neat because, first of all, you have to think, okay, on coral reefs there's a lot of interaction, but there's also a lot of, you need nutrients. So organisms need different nutrients. So some organisms need algae. It's a case of medium-sized fish, small fish, big fish, medium-sized fish. What you find is that coral reefs are actually like nurseries for a wide variety of fishes. And also, most of the things we eat. We eat crabs, we eat scallops, we eat lobster. You can all be found on the reef. So algae play an important role. This right here is, you can always, this is very interesting. First couple of times I've been to the reefs, I saw the large things. But as your eye becomes trained towards there, then you start seeing the little things. This is actually lettuce sea slug, species Eliza crispata. And it's very neat because depending on what type of algae they eat, that's their color. So you'll see these in a variety of colors. It's the same species, but depending on what type of algae they eat. And on the reefs, you have the green algae, you actually have the red algae, the red calcareous algae are actually more abundant on the reefs, at least at the facility anyway. So depending on where you are on the reef, you see different colors of these. And if you take them up in your hand out of water, you will be, your eyes will be blessed with colors you touch stuff, it'll come back. So, in terms of the coral reef itself, you have hard corals or hermatific corals, and these actually build the three-dimensional shape. At Salivia Bay, we have the finger corals, and Salivia you find these in patches. I've seen colonies as wide as this square right here. The only difficult thing in trying to map out the upper intertidal part of this reef is you don't want to step on anything. So it's, I have to train not only myself, but my help assistants as well, helpers as well, because I choose to use citizens to help me. In the pedagogy aspect, I'm in the Department of Curriculum and Teaching. Citizen science is huge with that. And citizen science is basically you take the common person off the street, train them, help them collect, they help you collect data, and I use that data, or scientists use that data to make an impact. Informal science basically is learning science outside the classroom. So I was telling Dr. Potts here that I had a high school friend and asked me, hey, my kids are coming up to Salivia Reef. We take some time to show them what's out there. And so, of course, because my high school teacher actually assisted me and directed me on a path of marine biology. So who's to say showing these kids, finger corals and telling them about symbiosis? You can't stretch 10-year-old minds too much, but you just light that little fire, that little one word, and that can make an impact. So whenever you have that, that chance, do it. Finger corals, Parietes, also come in a green morph. The most interesting thing about this is we don't ever see this. I have not seen this at Salivia, but only at the reefs next to the fishing depot at Toco. And I have to believe that I'm speaking to Dr. Philip at UWI and Dr. Toco, who's president of the Coral Reef Alliance in the US. Toco is an area where there's a lot of upwelling water, deeper water coming up. So that means that there's a lot of nutrients being brought up through the reefs. So that might be a cause of it. The finger corals, this is one of the few corals that actually extends its tentacles to feed. So not only do zooxanthellae provide nutrients from its byproducts, coral, the tentacles can actually get tiny copepods, tiny organisms, and actually feed from them as well. So notice they're like fuzzy, there's a fuzzy look about it. Those are the tentacles. This is the lesser starlet corals, terrestrial regions. And this is related because, lesser starlet corals, these actually look like stars as well. One thing about corals, when you see, so if you go to Boco Reef, Florida, or wherever, once you see coral species looking brown or beige, this is indicative of the zooxanthellae within them. All the other colors, like green or whatever, purple or whatever, those are actually GFP proteins that are reflecting the colors. So this, you can tell, for instance, I did some research with some genetic research, because remember, you have the coral animal, and then you have the algae, which is a different species. So I did some genetic work with Dr. Todd Lejeunesse. He's actually one of the top genetic coral symbiont analysis in the world renowned. His mentor actually, talked about classification of these symbionts. And one of the things we did was actually take these samples and apply the genetics to it and looked at the species level. So they described the symbiont species in clades or groups A through H thus far. And within A's, there's A1, C1, whatever. But some corals, you can tell what particular symbiont type they have within them. So like, for instance, Pseudoastrea will have like type C1. And we use that, science, we always like to want to know, educate us, we want to know. So, and we want to know so that we can share. So we're always diving, as more tools become available to us, we're always diving into them. We can get or receive and acquire more knowledge. One of the things you see out there during low tides is you see brown mats. So you have hard corals, you also have soft corals. So this is one type here. This is a zoanthid. It's also called Polythor, species Polythor. And what we find is that we find these more further out. These, believe it or not, Polythor, there's been some research that one of the drugs used for to combat the AIDS virus actually was developed from chemicals from these. You have the zoanthus, which is another type of soft coral. Here you see the orange. They're all the same species, but just different color morphs. You have this kind of bluish greenish. Then you have a brown. You know, you can name a carnival band, zoanthus, and have all these colors. Octocorals, up to 8, they have like 8 tentacles, and you really cannot tell the species name unless you actually look at it under the microscope. These are really someone from you did like an extensive PhD on octocorals of Trinidad. Her name is Lipson of the time. So you find octocorals, you find these bad boys here, phyllocorals, which actually is a hydroid. However, you know, if you have very sensitive skin and you touch one of these, you will already burn you, they're very sticky. Hence the name phyllocoral. Species name metaphor. All right, so with these, so I'm looking at the reef builders, the hard corals versus some of the soft corals. So on the x-axis, I have zones 1 through 4. Zone 1 is basically, and this is from some of the research done next to the fishing depot. Zone 1 represents 0 to 10 meters from the shoreline during low tide. Zone 2, 10 to 20. Zone 3, 20 to 30, and zone 4, towards 40 meters out from the shoreline. Percentage total. And 2008 is really represented by a gray bar compared to 2012 black bar. So if we look at the reef builders for the zones, you know, there's really no significant change really. These values here are not as significant. But if you look at the zoanthids, the soft corals, so this here is the Polythor, the brown, and then the zoanthids, which are their different color morphs. You notice that there is actually, with respect to 2008 to 2012, there are actually significant changes in zones 2 and 4. And so when we look at it even more closely, looking at the zoanthids, well, there's significant change or increase 2012. And when we look at the Polythor, we get the significant increase here. So thus, these two actually make sense here with this particular graph here. And that has, with the whole biodiversity and coral reef monitoring, these graphs here and these data here are very important to monitor changes over time. Now, coral reefs, I mean, 20, 30 years monitoring, you may not see changes. But if we have the data, then someone can't see, and this has been done, hey, Toco needs a port, so we need dredging, we need to do dredging and whatnot. And that has happened in the past, but the community came together and basically said, no, we cannot do that because we have some natural coral reefs, that's natural environments here. So the purpose of the coral reef monitoring is to have the data so that when outside entities come in, especially development entities come in, we can say, okay, you're going to destroy that, but this is what we have. So that's one of the purposes of coral reef data. In keeping with what we see when you land next to Toco Reefs, one thing you see is these beaded anemones, and this crucifer. And being Nigerians, remember, Nigerians share, for the sea anemones, this is like the collar. Notice the tentacles, and it has a radial symmetry. The mouth is in here. If you touch these, they recede into the hole because what you can't see is the column and then the base as well. And these come in all different colors. Really, I'm telling you, you could develop a whole carnival band from things under the water, right next to where you land. And the sun anemones, this one actually is toxic. So if you touch these will actually burn as well. Helianthus speculaculum. And by the way, these are all in during low tide, less than the high water. So if you can't swim in the water, you can walk up here. It's ready. The rock boring sea urchin comes in red as well as black. This is really interesting. So we know species mostly have, in keeping with the Latin taxonomic naming, two names, a species and a species habitat. So human beings are Homo sapiens. But this actually has three names. Echinometrel or something. So when you're doing your when your course will come up and you're explaining these students as well. But it's really neat that at the reefs at Toco, you see a lot of these. Like in zone 3 and 4, you see a lot of these compared to Salivia Beach. And these hide in a lot of rocks and crevices. This, the Caribbean octopus vulgaris, this is actually the only octopus species in the Caribbean that comes out during the day. And in March when I came here, it was really cool to see one of this at Salivia because I had only encountered these at Toco. It was really nice of this octopus to pose for 4 or 5 nights. Because, can you imagine when, it's like canister, oh my gosh, I can't believe I actually walk these things.
Wait, walk before you change off that, the behavior, because it's kind of like,
You can see it?
no, this is it.
Oh,
I've seen like what looks like one tentacle. No, I didn't just clear, just showing here. I've seen what looks like
Just
one
point
tentacle.
See the eyes? Yeah,
So the eyes are here?
Yeah.
The eyes are there.
Oh,
Separate.
right. Oh,
On the
yes, and what looks like
Yeah. It's amazing. When it swam away, it changed. It's cryptic. It changed color. It's like a white.
I'm glad I asked you for that. What I'm seeing exactly is it's an attack.
It's a mollusk, so
This is what I do.
final mollusk. Yeah, so it's a head foot.
Head foot?
Well, head foot mollusks. They basically have like the head that is like almost entire thing in your hand.
So it's not really.
This was actually-
It's really have 8 legs then. 8
It's
head.
Okay, when you say tentacles, you mean this entire thing here? Yeah.
Each of those.
That's how it goes.
Yes. Yeah, this particular one, it looks large here, but it was maybe when it swam from head to tentacle, maybe a 9 inches.
So you have to go real close together.
Yeah, well, I just happen to be in the right place.
I think
When you're on this is what I've found out. I have help when I'm doing my core monitor. And these are like, as we say in Trinidad, they're fat. So when they walk, they talk in and splashing. So what I do is I go first. And I take my time and walk. Because what I've learned over the years at Salivia or Toco is you never know what you're going to see. So I take my time, I do our reconnaissance first. And sometimes you see nothing, and sometimes you're just waiting on something, and something goes there. And I'm like, oh my gosh, I'm impressed with that. So this was really neat. So now with respect to biodiversity, this now I can safely say this has been documented, has been seen to 2013 at Selavia. So, keeping in mind this was readily, I think I saw a photo of this out there in the hallway. Yeah, we see these all the time. It's a brown and yellow eel. But what most people don't know is like they think, oh my gosh, when they see this, they run the away. But these are actual, they look ferocious, but they're actually harmless. They're really harmless. So you see, yeah, the most people think it's a snake, oh my gosh, and a minute. No, not these here. This was actually, but I'll be honest, the first time I saw one of these, and you see these all the time, is I saw something twirling in the water. I was like, that can't be a snake. And then when I saw it, I realized it was an eel. You know, I flew out of the water. That was the first time. And then I read about the species there. And eels are generally not, I mean, you don't have to stick your hand and harass the eel for it to bite. But this one let me come within this to take enough food. My hand is there. So they're harmless. They really are. I can't convince you. But they really are. This actually looks like something from sci-fi. First time I saw this at Solidarity last week, it's a sea cucumber. They're small. At least there's a green version and a more burgundy version here. Sea cucumbers are really interesting because if you pick them up, they'll slide all the way in hands. I mean, red gooey slide. The larger ones in the Red Sea, for instance, I mean, if you harass it, they'll slide all over it. This is the things male divers do when they have free time. They play with sea cucumbers. And they're actually, it's very interesting, there are actually some species that a type of, it's a shrimp or thin fish actually goes up, not this species, but goes up in the rectum and like lives in there. And it's not parasitic, but it comes up. National Geographic documentary shows you would be amazed at the things. But this sea cucumber, so it's documented now that it's the first time I've seen these, and this particular one was found like 180 meters towards off the shoreline. So once again, further you go out, the more biodiverse. So that's kind of part of the things that the American Museum of Natural History, my proposal to them, really is to document the upper and tidal reef. So kudos to that. This is really, it's a type of polychaete worm. First time I've seen this as well. Phylum Annelida. It's a segmented worm. And this makes a great Instagram pic. This is so neat. You just can't imagine. It's just really beautiful. These are basically, we saw documented 12 of these, some 12 centimeters long, some longest one I think was 18 centimeters long. I'm not sure what the species, I had to do some research on that, but I thought it was really neat to actually get like really close shots. So, coming towards the end, this is alpha. It's a green algae, but it's actually, when you see these growing, it's actually a sign of nutrient input. So on the reef flat, if you're lying like close to where the river is, to the immediate right is where most of those organisms that I showed you, we took photos are, from there. However, so if you go around the corner where you can't see nothing, we see a lot of these growing in between the corals. So there's some level of nutrient input in there. And that can be caused from fertilizers, it can be caused from a lot of different things. To pinpoint it, you have to do some more water testing or whatnot. With respect to, if you think of the coastline reefs of Trinidad, remember we just hear about Toco Reef. Toco Reef is actually naturally, the government is a protected area, but there is no protected area for the only fringing reef in Trinidad. And this has only been slowed up because of land dispute around the area. If that land dispute ever gets resolved and people start coming and building, I fear that we will lose this aspect, this natural resource, the old fringing reef. And so, with respect to no drinks allowed, the no drinks allowed part is basically the human impact on the reefs. Okay, you've heard about coral bleaching, which is basically the tiny algae that lives in the coral. They leave the coral, and the coral becomes pale white. And so when it becomes pale white, then the fishes, they leave, and then everything else leaves. And this has been seen more likely with rising ocean temperatures. And not only rising ocean temperatures, but prolonged rising ocean temperatures for a long period. Certain corals, species, can withstand longer levels of rising ocean temperatures than others. A new trend we're seeing now is ocean acidification. With the carbon dioxide input into the atmosphere, simply because, okay, if you want a Hilux or an SUV that puts in more carbon dioxide to the atmosphere, who's to tell you? Does anyone want a Hilux or SUV? Who's to tell you to buy a hybrid or, it's the carbon dioxide, most of the carbon dioxide that we pump into the atmosphere, where does it go? It goes into the ocean. And so it goes into the ocean, it becomes carbonic acid, and so it's slowly and slowly losing the acidity of the oceans. And when we think of coral reefs, calcium carbonate, when we think of a lot of mollusks and a lot of gastropods with shells, okay, these acids basically can disintegrate these carbon dioxide. So we're looking at a trend here that will more than likely, if we don't control it, impede some of our natural resources. Sediment input. Once again, if that land dispute at Sylvia ever gets resolved, then the development, once you develop and build resource and whatnot, the sediment, all that dust goes into the rivers, which pumps well into the reefs. Now remember, corals, they have the tiny zooxanthellae in them, and these need light, sunlight, for photosynthesis. So that, the smothering of the corals is detrimental as well to the reefs. Nutrient input, once again, fertilizers, chemicals, you name it. In Finding Nemo, Gil the pelican says, you know what they say? All drains into the ocean. Well, when you drink your cup of tea or your coffee and you don't finish it and you throw it in the sink, guess where it's going? It's going in the ocean because all drains lead to the ocean. And that's not my quote, that's actually some of the top coral reef analysis. These are their quotes. Alright? And one final thing is that overfishing in the Caribbean, we're having an influx of landfish. The landfish, once you overfish, you take away the groupers and whatnot, what happens is that you take away a top predator. The landfish, which is an exotic, doesn't have no predators or whatnot. So, and it's very voracious. So they are wiping out many fish species. And that's actually making its way down. I think, I'm not sure there was a sighting in Tobago, but if this ever gets down to Trinidad, it will cause havoc on our fishing industry. And a lot of these local communities, especially Toco, depend on these fishes. I don't need to tell you how expensive fish gets during Easter, right? This is detrimental. So next time you're climbing next to Toco reefs, think twice. Keep your bottles in. Tell someone, hey, don't throw anything around. Don't destroy your habitat because it's our natural resource. And tell them, make sure and tell them, no drinks allowed. Any questions?
Well, before we take questions, I would just like to make one comment. The word that comes to mind is grateful. I'm very grateful for it. It was wonderful. I enjoyed it all. I was touched by your references to the impact of teachers in high school. That's not because myself, right? I never taught in high school. But because I do know of teachers who have had that kind of impact. As a matter of fact, I wish I had that kind of impact. I wish that in environmental studies or environmental science and management that we had more of this in the field possibility. When we do take our field trips, unfortunately, it's the factories and quarries. But in biology, they can, you know. But my next question was, all of these beautiful, beautiful pictures, because I found this presentation so soothing, and I need it because I feel stressed. So sitting down here and watching all these things in the ocean, I kept thinking I can see this, you know, in like a little mini museum, or I think this has to go to the National Museum. I think we should get a little reef room, but I don't know if you're allowed to.
Well, I can say, to answer that, I can say over the years, what I do is these photos, I take them back to Auburn, and I identify the organisms, and then I make a poster, and I laminate it, and I donated them to Toco radio station, BITS, internet cafe they have there that's never open, the Keyshawn Walcott Lighthouse next to there's a arts and crafts center. I've donated it to there, and it's never open. I've never seen my posters in there. I have seen they put one up in the lifeguard, and the guy there said, "Man, just bring them, put them up," because I've donated to Toco Composite the copies of my thesis, masters. Yumi has one. It's a slow, yeah. But if
I think about the visual impact this has, first of all, as even slides, but as something moving surrounding you, permeating the atmosphere. In addition to which, when you leave it in Toco, your picture can be converted. You need to leave it in Port of Spain, when in Port of Spain or San Fernando, these cities know what Toco has. I never knew we had it.
Well,
you know, we are my smirk at this, but the average person may not know.
You're correct.
But in some cases I avoid running the average, let me be clear. But I did not know that we had reefs and Toco. Okay? I mean, in the case of a lot of Port State people, Toco is Michael Henry, Carnival Sunday, or Carnival Thursday, so they get
hence the title.
Yes, I know. You know?
I think the first step really was making the connection and letting you all know I'm more than happy and willing to share the knowledge because it's something I want to do.
Well, not to touch me, this emphasis on wanting to share knowledge. But you know what, you see that how you have these, I would like to see these things around me, creating an atmosphere, a little room. What about down by your own? And it's such nice, I don't want to say scenery, instead of having into that UTT thing.
I do not always but I just doing it up there.
I would like that.
So this is how actually have suggested ideas or having a projector or so many
maybe Dr. Potts, we can collaborate where I can send you the photos.
I was very soothing. And I think we need some soothing.
It'll be nice to have a marine biology class and all you see on the wall.
Well, the thing to it, and Professor, remember, as you see, we just getting started. And usually when you get a number of active, excited students doing their work, you tend to find people willing to set up maybe a marine aquarium here or one day and be able to maintain it.
Those of us who are
not 2 years, they are doing a master's here.
when you spoke about bringing in public citizen science,
Yeah.
one thing you didn't mention, when you said that, you take them and you train them and you collect data, what you also do is you leave on them a larger awareness of the environment. You see, like how you mentioned, they splash and they this and they that and you walk before. Well, they pick up on that. And all the people who help you and things like that, when you leave them far more aware than how you found them. So that is a second benefit. That is the gift that keeps on giving.
Right.
Because then they pass it on. But to come back to my original request, I would like to have this spinning around me, soothing me on a daily basis. I'm not going to get my office and see what it is.
Eventually she'll see some of those. When we have the students involved and they're willing to get started, put it on maybe some period, then it's fresh water and also water, and some people just take pride in doing that and see the joy that others get over. Alright, because it will come from a little bit of which they can, but you students do well. And so that will happen. But getting down to a little bit of what they talk about. So you're monitoring abundance and distribution and to what end?
Just
Well, I just want to tell you.
well, so just from an annual perspective, so I'm looking at our corals. What you're supposed to see is, for instance, in the Caribbean, the most with respect to the abundance. I think Jamaica has 20-ish%. That's like the highest in the Caribbean. So about 20-23%. Like with respect to abundance, abundance.
Should we get a little bit more happening in the beach? One or two of those we still. We still have. There's some work done with La Pointe and others, included with Reefs some time ago, and we did some monitoring of all the little reef plots, so to speak, around the beach. And there are some of them that actually show reasonably high percentages of hard coral still.
Okay.
Well, of course, some little soft coral, but
but
that was the go back in the 2000 time, right? So Tobago still has some, right? And in fact, would you have found it interesting at all in doing your work here to have actually done a little bit more on the nation's reefs? At least something in Tobago to compare with what they're doing in Toco to society, but told a different story.
Yeah, I think as an avid scuba diver, I've tried to, I think Coral K, they're out of the UK.
Yes, they are.
I've tried to make contact with them to set that up, but my love is up to,
But if you have talked to Booker Reef Trust, at the time they still had a vibrant thing going at Coral K. Was it me working with them on some other two projects? And you may have been able to do a little bit more of that. And as I said, like Professor, let me congratulate you for doing work on Toco to start, because a lot of people who at least know that there's still a little reef out there in Toco think it is insignificant and doesn't need to be worked on, and that is not true. It is important to still do something, and in fact, you have been able to show that things are happening, and you say you have two peer-reviewed people.
Yes, so this is...
And which journal
this is...
biological sciences, visual biological sciences. And the other one is what?
So this is I mean, 2000, it was the initial, this is one of my thesis. So actually both of these are all my thesis. And then we also did some water quality stuff. I think I got in contact with Dr. Philip, and we're going to co-author another paper on that, kind of connecting the physical attributes to the distribution. Because you have to do that as well, acidity, temperature. You have to connect water quality as well with, the...
Oh, you should do. Maybe we could have done that for you next year. Because as part of the environmental science and management projects, this year we have 27 program projects. So quite a few of them are looking at water quality and connected with land-based pollution. But we did have a couple of projects, and Sasha, who is one of our soon-to-be graduates, sitting next to another one of our, so those are two of our environmental science and management students, and Sasha looked at oysters in brackish water, and in 3 different spots, and we looked at water quality. Well, we were already looking, they were already looking at heavy metal pollution, but they also looked at the water quality parameters, because that's essentially mostly what we do, what we can do.
And
It's not in the
that's very
science.
important.
Yeah, because those are the meters we have to. So even to get heavy metals done, we have to do a lot. But what we have also started doing, which is what Jenny did, is looking at doing a lot of socio-environmental surveys. You know, and bringing everything together, the socio-environmental, the water quality. What we can't do unless we have the input of a biologist is any kind of species investigation. So short of doing heavy metals in a matrix is not species investigation. You know, we don't do any biodiversity and things like that. There really is not enough room. There's no opportunity to do it in the program. It would be nice to sort of,
come back with that.
have some sort of course on it.
Well, it's always food for thought,
Yeah, well, one of the things that we go to,
But I think next thing we have to do.
one of the additional type of research thing I want to do, and for the job, I think we mentioned earlier, and if we still want to look at what else we could collaborate with on this side, especially now we get this side of the whole thing, right? So it then not just be a UV focused thing. As Professor pointed out, we do have people here working on different aspects of the marine environment, and so on. And of course, once we get the thing going, we can have our own people and students involved with different things that we can do.
Yeah, I think I was really excited when I saw that program about to start. I mean, that's one of the reasons why I wanted to connect with you, because I've not seen anything like that offered.
It is different.
Yeah.
It is a different.
I know ecosystems are in the high school program, so it's bewildering to me to have a high school and you can hop, skip, and jump to a core ecosystem, and teachers and students are unaware, oblivious. It's just bewildering. And it's been tooth and nail trying to get.
Yeah, the training has not been.
Yeah, I mean, I've spoken to principal, teachers. It's just hard to get there. But I'm not going to give up.
You have to get it on the Cape syllabus first.
Anybody else have any questions?
Science component on the Cape syllabus.
Yeah, I mean,
But I don't know I don't know how much biodiversity, but they could do that, because I think they do an extended project.
Yes.
Like somebody had to do oysters in the Caribbean swamp, oysters again, measured water quality again, so they could do it.
Well, Dr. what his name is, Davis, was saying to me that he was now helping to develop a key agricultural science curriculum.
In UB?
No, the CMC people.
Oh, okay.
Yeah.
No, but there is an environmental science study, so this is what.
Oh, the environmental science is already there?
Yeah.
Okay.
Yeah.
But they're actually doing an agricultural science one towards well right now.
Yeah.
So they may very well find that more teachers may expand their horizon and get more involved with the marine sciences.
Yeah. I mean,
as they do,
it's a
And curriculum development,
Yeah.
which is what you are interested in.
I've just, go ahead.
Sorry, no, I was just going to make a quick comment that we also, in marine sciences, have actually started going
This is
to
not
studies.
Duffy.
Okay.
From marine sciences.
Been to 2 schools already in South, at least, and they're interested in having marine biology clubs in the schools after we had presentations on programs and
And
things
invite
like that.
students
So,
to come.
yes,
Yeah.
so it's like at the start.
And that's
for
actually
the schools.
a training ground for the graduates here to give like guest speakers guest speeches to them as well. I mean, you can actually use your students here. Because undoubtedly, I mean, what's going to happen is, you know, the students here, I'm still a very shy person, but for me to share the knowledge, I have to stand up and be comfortable in front of people. What you see in Trinidad and Tobago is that a lot of people don't know how to speak. You know what I'm saying? And you can transfer a lot of knowledge if you're able to communicate better. And they train us. Yeah. We have Fridays, we have Kusan, we have Wednesdays. I mean,
that's all we did, yeah.
And we're encouraged to publish as well as PhD students. I mean, you know, which I found out here, you know, there's not much. I mean,
Young University here, we're moving fast. Anybody else, any more questions for Mr. Stanford?
Mr. Belford.
No question? Sarita?
yes.
No, I actually just wanted to find out if you go in this direction and any of the organisms that you as key organisms then as indicated species
Ooh,
for any particular political stage. Because I would like to build on that
Yeah.
in terms of looking at how humans impact the
Right,
ecosystem
right.
rather than just
Right.
looking at reality.
It's interesting to say that, yes, if I had the funding, I would have maintained the genetic aspect, because with the genetic aspect, you can monitor the symbionts within the corals. And you can actually, when they bleach, right, you can actually monitor what species come back. Because there's an adaptive bleaching hypothesis. As we're moving towards, there's a lot of environmental change, and as we're changing patterns in the climate, some corals, or some scientists believe that corals will eventually evolve. And so we're seeing that research has been done where they're seeing that bleached corals maintain the D1 symbiont because it's like adapted, it can withstand bleaching. And we don't know why. I mean, it's really exciting research. So with respect to like an indicator, I'm excited at that aspect. I guess with respect to fire corals, those are the first to bleach.
Have you started to look at how you compare like the I don't know what to say, the population communities now compared to what was done probably back in the 80s? Remember when I was talking to you about?
That'll be a nice publication.
Yes, I actually.
I'm always thinking publication.
Because that's what I'm looking at. I would like to compare what is done back in the 80s to now. What is your finding? There's a big gap between then and now, and see how the distribution has changed. And that way we can find out whether or not the human activities here have impacted on the distribution.
Right, exactly. That's
Speaking of that, if you do that, you're going to have to be statistically. Yeah, I know you put up your bar charts. So did you do any statistics?
Yes, actually, I did because I marked my transactional GPS. I think I started with in terms of species, I did the Simpsons Index, and in terms of the percentage, I did like just basic T.
Yeah, because those bar charts you put up in
I should have indicated like star to and the
well, you said that in the second, what would you call it, not section, so one of them from, let's say, 10 to 20 meters out. And for the second and fourth zones, yes, you saw significant changes because 2008 and 2012. And I assume
yes. I should have done correct. I should
that you had done, probably you would have needed to appear data t-test.
Yes.
But in terms of this, looking to see the distribution has changed. That becomes a little bit more complicated. You know, if you're truly looking at the distribution of species, but still doable. You know,
when I began,
just don't do it in terms of percentages.
Yeah, when I began this, it was difficult to get data from that. Like I couldn't find any.
You will have to count on your own data, which is why it is important to continue monitoring. I know you think Tobago, Toco is a little reef, Dr. Potts comes from Tobago, you know. And so, you take Toco from here to Toco. He was noted, but I noted. But on a different note, you said if you have money, you will maintain genetics, genetic material monitoring, let's say. You know, this is the thing now, because on a different, just to digress a little bit, we have lost our buffalo DNA. You know, we have buffalo, Trinidad has a unique kind of buffalo.
Bison.
No, well,
But we call it our unique thing.
but the buffalo was the original thing was done by somebody. I forgot the man's name. It was a shame because it was but apparently there were numerous advantages to the meat, and it is the DNA was sort of like sent out. It wasn't patented as it should have been. I was sent out to different people, so now they are buffalo herds built on our DNA all over. In the meantime, our buffalo is dying out. There's brucellosis or something which is killing off some of them. And every time that happens, you have to shoot that and put that down. And the whole thing is, all of this would have been prevented if we had kept the DNA back. So I think the government understands now, but certainly the universities do, that keeping DNA banks is very important. It's one way to sort of, in a sense, protect your biodiversity.
Tissue banks.
Right. So when you say if you had the funding, where would you maintain this? Because again, I think that I know that the PhD is consuming, but I think that on the other hand, every time you come down here and things like that, perhaps there's somebody you need to talk to in the government.
I've been told that, on this trip I've been told that.
Yeah, I think so. About first of all, about getting my little mini museum. But secondly,
No, it makes sense.
if you think you want to keep a data back, because you know, honestly, a little mini museum here, even in Toco, where people go and you say, going to the little Toco museum, see what they have and say, hey, you have
You had something like that in Toco. At the initial Imperial College Agriculture thing, yeah. And then we in UTT have been talking about something in sugarcane.
I see Dr. Guppy has something, she has a
something sugarcane gene bank as well. And we have something to celebrate.
We need to have a gene bank.
Yeah, so we have some surpluses of some things here.
I mean, what we do now is, what I do now is, I just put them as a voucher in the museum. Classic example, like this, I was on Twitter, and they were doing a deep water dive in live, this type of phenomenon in the Gulf of Mexico. And so on Twitter, you chat with scientists. So I use Twitter for my students, because I teach science. And Twitter is like, they're always like, tell them you can be lying in a bar or in science. So I was able to, from Twitter, communicate with a scientist and acquire 2 deep hydrothermal vent shrimp from almost 5,000 meters through Twitter. And so I shared this with the brand new museum we have in Melbourne, and the guy, one of the curators, his face was like, well, you can do it permanently, to have it. And so I was able to do that. So there's always one there, and then I have one for, I do a lot of elementary work, middle schools, high school, elementary school. It's cool to, hey, here's a shrimp from almost 5,000 meters, in your hands, that I got through. I look for it, because I would be curriculum teaching. I bring my corals to class. Even when I'm tutoring, if I'm tutoring Wilson's, I bring my corals, I bring my anemones. Because visual, there's a lot of visual learning.
That's what I was telling you.
That's why I threw in a lot of schools.
Just a couple of things that will actually help you as well. The Palethora arborea, it's also indicative of nutrients.
Okay.
So the higher percentage, so to speak, though, the more that you find it
Yeah.
on a reef system, it's generally is a loose field indicator of high nutrients in the environment. So if you see lots of that, you know that there's a lot of, not necessarily from runoff or anything else, but just naturally from within there. You had also mentioned something with coral bleaching, and you had your particular stress was on raised temperatures. Coral bleaching as a whole is a natural phenomenon that can happen with any stress.
Okay.
Right? So whether the temperature
So you can cool,
drops
yeah.
or high sedimentation, it can actually elicit a bleaching
Right.
response as well. You said something with the lionfish. We actually do have lionfish now in Tobago.
Yes,
It
I
has-
read like one
yeah.
report.
No, it was even cited just last week. I was on a dive and cited some there. And the unfortunate thing, certainly within the Caribbean, our group of species in the Caribbean don't eat the lionfish.
Okay.
Because of the type of species that eats the lionfish comes from the Pacific side.
Yeah, it is. It's
Yeah, we have,
We should do like what they do in Florida, have scuba spears and.
they do that actually.
Okay.
There are a couple of things that they are doing. They do spearfish just to kill them. And there's also, they have small pockets of persons have started trying to catch them for aquarium trade. And then the majority of persons who do kill them also to catch them are doing it in a movement in the Caribbean to see how best they can cook it. So there's been competitions throughout the Caribbean to cook the lionfish because we eat everything.
Yeah. That's good.
There was the quality worm that you had seen in a much of the farm was quite common in our waters.
Oh, okay.
And they do have different colors, and if you go up near them and they are startled, they actually get the white bristles. They change that color, so they just like bristle up like
So
flowers.
if you took a pencil, it would be like touch them and it just shaders so cool.
You don't even need to touch them, you can just actually, this, from these shadow movements, you can actually elicit that response as well. And the last thing was like, there's one of their photos, and they were fantastic photos by the way. I would have loved to have a scale on some of them at least, but that's what we're lacking, of course, particularly for Toco. We have a lot of things that gets hidden in people's personal lives, and so having your photos in a public setting is fantastic. Just one particular species that you had indicated was the Siderastra aurelia. It was actually a solanastria in there, so it just needs to be a little bit, it's sometimes difficult.
Yeah. No, there's Siderastra.
Yeah, there's the citizen and then the siderads, but
Okay.
that particular species that you had showed, which is also visually closely to citizen, is a solanastria.
Okay.
Just as a
Is there a green morph to that? Because I've seen a green, but not like the way star.
I've not seen a green one to that. There's another, lots of other
Yeah.
species in there.
Because
So,
I've been like that, if where the lighthouse is, like to the left, you can go down, there's no path. Because I'm always searching for new sites. And I think that's where I saw the coconuts and the fish.
Right.
The
Yeah.
the juvenile.
And then the last comment that I had was with the zooxanthellae, and you were talking about some of these genetic studies and these sorts of things. I have 2 points to make on that particular one. We have, well, when I was doing my PhD a few years back, collected from certain species in Trinidad because Todd Lajeunesse, who you had referred to, Todd collects, well, as you had mentioned, he's one of the most well-known persons who does work specifically on zooxanthellae. And I was fortunate enough to meet him a few times. And he wanted some, not just from corals, but on anything that may have, zooxanthellae. So he actually has already some samples that have been sent to to Bill, who was one of his
So here in his lab, he's moved now.
Yeah, that's quite common as well. But the thing of it is that in UTT, we have projects available for students wanting to examine these zooxanthellae so we can extract them from whatever the species are, raise them, so to speak, and then be able to do the genetic studies as to what we have. In addition to that, we also have our water pan, which I don't know if you're familiar with the water pan. But with that, we will be able to, in our studies, to look at the efficiency and effectiveness of these zoots that are on our reefs, as well as any other marine plant species that we have. And we, so far, that we know of, and not an extensive amount of work has been done, locally we don't have the D strain of zooxanthellae. That's something, again, and most of those studies have been done primarily in the Pacific from those species, and a lot of the database is biased to that. And it's something that I'm hoping that we can get students, multiple students involved in doing that and coming up with a fingerprint for our species, both pre, post, and during bleaching.
Because I tell you what, the Caribbean has 175 species, whereas coral species compared to the Pacific, 400, but we have more diverse symbionts. We have a range from A through D, E, whereas the Pacific mostly C, type C.
Yeah.
So we actually have a good reason to monitor that as well.
Well, actually, not just to monitor, but even just to even like go and identify,
Yeah.
because we don't have the concerns. Because you find that you have multiple strains
Yes.
of what we do have that we don't really know. And again, it goes back to these genetic studies.
All right, guys, I have to jump in. That just, you know what I just said then? That you finally found the coral reef biologist at UTT, and you only to find your own time and continue this beautiful conversation, which I'm sure will bear fruit to both of you as Trinidad and Tobago citizens. Ladies and gentlemen, since you have no more questions to answer, this wonderful seminar is adjourned. Alright, so started, this is the area I've been telling you about all the time. So you and Dr. Lopi, or you have to find time for lunch, because she is our story. Yeah.